And time to start. So very welcome to today's webinar, where we will investigate the mechanism of T-cell infiltration and survival in breast cancer. Today we are two presenters. So I'm Ann Holmberg from Qlucore, where I work with training, support and sales. And
I'm Peter Revel from Clarivate, where I'm the product manager for MetaCore. Anne, you're on mute.
so something happened there. So so the agenda for today is that we will start with an introduction and give you a little background for around 10 minutes. Then we will do a live demo of the analytical workflow using Qlucore Omics Explorer and MetaCore for 20-25 minutes. And we will end with a Q&A for about 10 minutes. And then you need to use the chat to ask questions to us. So this will take a maximum 45 minutes. That's the plan. So let's now quickly start by introducing the two softwares to you. So Qlucore Omics Explorer is a very fast and easy to use tool to analyze and explore data without being a statistical expert. And the software has a lot of functionality. For instance, we can visualize your data, and that can help you to find outliers, to detect structures, new groups, and maybe generate a new hypothesis to find new things in the data. You can also analyze data using a number of different statistical tests to help you find discriminating variables, and you can present your results in a number of different plot types. The software includes gene set enrichment analysis, as well as, just close that little window there, possibility to build classifiers using the built-in machine learning algorithms. Today we are going to concentrate on this functionality to identify interesting genes for the relevant for the tumor infiltration that we're interested in, and then present those genes in a couple of different plot types, and finally export them to MetaCore for further exploration of the underlying biology. So that's the plan. And now a brief introduction to MetaCore, which the slide comes here.
Okay, so as Anne said, MetaCore is a database for exploring the underlying biology behind your omics experimental results. Over on the right-hand side, Metabase is the core content database that sits under the hood. It is a molecular interactions database. We have almost 3 million interactions there. And an interaction is just a straight interaction between molecule A and molecule B. So in this example, the CD1 interacting with the T-cell receptor. Each interaction has an interaction type, so in this case B for binding, a direction from CD1 through to the receptor, and an effect. So the effect would be, for example, activation, inhibition, and so on. And the entire set of interactions of nearly 3 million have been curated by Clarivate scientists by reading the literature, the scientific literature, and pulling out evidence for the interactions there. So it's all supported as well by the source data. Another type of content in Metabase are the pathway maps. So these are manually curated canonical signaling pathways that are curated into a single map, and they'll also be relevant to today's talk. Over on the right-hand side of this slide is yourselves, as users, or myself today giving the demonstration, inputting a query. So the query can be as simple as a keyword search, a browse to look for the content, or it could be what we're going to be doing today, which is using omics analysis, so uploading data from an omics experiment to query the content. And MetaCore is that query tool that sits on top of Metabase with all of the algorithms that are needed. Okay, so in the next slide, we've introduced now the two tools that we've used, the Omics Explorer and MetaCore, and now the introduction to the topic. So this is about tumor cell infiltration by lymphocytes. So the immune system cells do infiltrate into tumors, and that's part of the process to detect and eliminate and clear the cancers as they start to establish themselves. So a normal immune response involving killer cells, dendritic cells, T-cells. Today we'll be focusing specifically on the CD4 T-cells and the follicular helper T-cells from an experiment that was run using differential gene expression, looking at the expression of the T-cells as they infiltrate the tumor. The relevance here is that tumor T-cell tumor infiltrating lymphocytes have been associated with a better overall prognosis in breast cancer, and also a better prognosis associated with the chemotherapy when the chemotherapy takes place around the time of the surgery to remove the mass, the tumor mass. So better prognosis involved with infiltration, T-cell infiltration into the tumor. Okay. So the next slide shows what we'll be demonstrating today. So Anne will, we're using a data set that's publicly available, large set of data of differentially expressed genes that Anne will then upload into the Omics Explorer and demonstrate identifying signals in that large data set. And we'll be focusing on those that have a low infiltration of T cells and those that have high infiltration of T cells, and looking at the differences there. Then Anne will export that the interesting genes, the ones that we want to explore, in MetaCore ready format, and then I'll take over and import them to MetaCore and go through a pathway enrichment process, a network building process, identifying potential biomarkers and even a possible target. So demonstrating the utility of MetaCore to explore the underlying biology. So this next slide gives some details of the experiment, some background of the experiment. So this is, as I said, the data set is publicly available from the Gene Expression Omnibus. It is a differential gene expression using Affymetrix microarray platform. And we'll be comparing tumor infiltration in breast cancer patients with a low level of infiltration versus breast cancer patients in a high level infiltration. So the objective today is to look at the difference between low and high level infiltration, look at the differences in the gene expression of those lymphocytes, and ask, what's the difference between these two? Why is one set of lymphocytes driving into that tumor mass, activating a response and contributing towards better prognosis? And why are other in different patients, is there something that could possibly be going wrong, that it's not activating that immune response so well and leading to a poorer prognosis? And then the details of the fold change will set the limits, and will display that, show how to do that, and set the false discovery rate limit as well. And then here for your reference, and we will share this slide later, is the publication associated with this study. Okay, so we've given the introduction, and now over to Anne to begin the first part of the live demo.
Yes, thank you, Peter, for this interesting background. So let's move over here. So we are now looking at Qlucore Omics Explorer, and I already uploaded the data set here. And you can see here that we have 34 samples and 54,000 probes corresponding to 22,000 unique genes. And we can also work on the gene level if we want. So what we did is that we downloaded cell files from Geo and then imported them and normalized them in Qlucore. So we are looking at the data in a PCA plot. We are looking at, so each blue sphere here is a sample from a patient. And we are looking at data in 3 dimensions, which is an excellent way of viewing your data, even if you have many, many more dimensions in the data, which is the case here. But we are looking at the 3 dimensions where we have the most information, and that's very, very helpful. So let's have a look at the annotations that came with the data set here. So we have no, sorry, I wanted to start here. So we have 3 different types of samples. So there is, sorry, I think I've taken away something here. No. Yeah, we have 3 different types of samples. So basically 3 samples from each patient, from lymph node, from peripheral blood, and from tumor. And let's color according to that. And then we can see that the tumor samples are different from the other samples. And the reason that I say that is that the tumor samples, they cluster together in their own group. And so samples that are close together are similar, whereas samples that are far apart are different. So we see that this is a very clear group and this is not as clear. But by doing an F-test, for instance, which we call a multi-group comparison, we can see if it's possible to find discriminating genes for the different groups. And by filtering down using a statistical filter, we can see that yes, it's possible to separate all 3 groups from each other. And here we can also see that one sample among the blood samples is a bit different from the other one. All the other ones cluster together, but this is different. So then I can use the label tool to find out which sample that is. And maybe I know something about this particular sample or this particular patient that can explain why it's behaving differently. And then I can clear that. And we also have information about T-cell infiltration. So we, in these patient groups, we have patients with high T-cell infiltration in the tumor as well as low T-cell infiltration in the tumor, and a couple of controls. But right now we will just take away the controls for the moment. And if we color, we can see that we have all types of samples are represented in both the high and low for the high and low patients like that. And now we will focus on the patients with the high infiltration. So we just take away the other ones for the moment. Like that. And we will also focus on only the tumor and the peripheral blood samples and make a t-test, which we call a 2-group comparison on type. We want to compare the tumor samples with the peripheral blood and see if we can find discriminating genes here. So by just dragging the statistical slider here, I filter down to a Q value of 0.01. The Q value equals the false discovery rate. It's the corrected p-value, so that's the one we want to use. And I also want to add a fold change filter. So now I can type a value in. So I'm focusing now on genes that are very good at separating the tumor and the peripheral blood samples, and where there is a twofold difference in expression between the two groups. So here we are, and we can save this result as a list. So let's just clear some old lists that I have here. So I make a copy of the active list. I give it a name, so high and fold chains equals 2, for instance. And I might want to add more information to the list. I might want to see the gene symbol, for instance, the p-value, the q-value, and maybe also the fold change, like that. I have all this information in the list, and I can sort it on any column by just clicking on that column. Okay. And then I can show these genes in a heat map. So I open up a new synchronized plot, like that. And here I have them. And let's go to the View tab to configure the heat map. So I press Auto and Auto to show everything. And I hierarchically cluster like this. And then we also color according to the type. And we can see that we have upregulated genes for the tumor samples, and we also have genes that are upregulated for the peripheral blood samples, like that. And I can zoom into the heat map to also show the gene names. Let's change to that, and then I can scroll down like this, and when I want to show everything again, I can do that. Maybe I would also like to see this information in a volcano plot. So let's open a volcano plot, like that. And let's configure it a little bit. So we have a fold change cutoff of 2, and we want a p-value cutoff of something as well. And maybe we want to color on fold change, for instance, like that. And this is also dynamic. So if I change any of the filters, you see that now I put back also genes with a lower fold change, and then I go back to 2 again here. And I can see that I have a couple of genes here with very high fold changes and very, very low p-values. So let's check what they are. So I just use the label tool and circle them. And then we change the label so it's easier to understand what we have. So here we have the gene CXCL13, and we also have the gene FN1. And there are 3 or 4 probes actually referring to the same gene here. And maybe I would like to have a closer look in a box plot to see how these genes are expressed in the different samples. So let's open up a box plot and put type on the x-axis, and I can then actually select from the volcano plot that this is the gene that I would like to have in my box plot. And here it is. So let's just change the label here on the y-axis. We see that it's actually that gene. Yes, it is. And then we can include the data points here to have a closer look, and we see that they are very, very similar in their expression. And I can also show other genes. So that's so, and then we just go down in the list here like that, for instance. So one or many, if I press down shift, I can select many. So if you want to compare a couple of genes in the same box plot, you can do that. And so now I'm ready to export this to MetaCore. I will do that, and then I will repeat this, and then instead go and do this same workflow for the patients with low T-cell infiltration in the tumor. But now to export this to MetaCore, I just right-click, copy to clipboard, MetaCore symbols, and then I go to Excel, paste this in, save it as a tab separated txt file, tab delimited txt, and call that high fold chains equals 2, for instance, like that. And I save it. And then I go to this menu falls down, but I actually have so here. Then I go to MetaCore, select upload here, and then I'm ready to upload my gene list here. And this will now be shown how to do by Peter. So over to you, Peter.
Okay, thank you very much. I'm just going to share my presentation. And I think you need to give me presentation.
Sorry, yes, of course. So let's just participants and give you this. Yes. So
Okay.
you are now the presenter.
Share my screen. And here we have MetaCore. So over on the left-hand side here, we have the toolbox of all the different algorithms and workflows that I mentioned earlier. Today we're going to use the upload to show to upload the set of data that Anne just processed in Omics Explorer, and we'll use workflows and reports as well to compare the high and the low infiltrating data sets. Over on the left-hand side here, we have a panel with other experiments, various different data sets that I've uploaded in the past, and a panel with activated data sets ready for analysis. And as you can see, I've already uploaded this data ready for today's presentation. But I'll go through first the upload process. So clicking on this upload button, and then today I'm uploading an experiment with gene IDs. So I would come through here and just follow the workflow wizard. So you can see here that various different files are accepted and they're recognized and matched with the content that's in MetaCore. The format is explained on how what needs uploading, and today I'm going to choose the file, come across here to this one which Anne just prepared, and I'll open that. You can see it highlighted here in this field, and I go through to the next step. Okay, so here's a sample of what it would look like as how it's recognizing the files. So you can see here these Affymetrix IDs in the first column, and the fold change in intensity in this second column here, the log fold change. And it just asks me, is this correct? Am I happy with it? If so, go on to the next. This is a study using human data, and I would go on to the next to upload it. What happens now is that this will upload and open a dialog box to process the data. It indicates when it's completed, and then I can close that box and I come back to the start page. And I would normally refresh that page just to show the newly uploaded set, but I've already done it earlier. I've got these ones that are activated here, and I'm ready to go. Let me just give a quick refresh here. Okay, I'm going to remove that second one that I just uploaded just to get rid of that. And then we're left with these two sets, which I now have activated. So if you remember, one is from patients with low T-cell infiltration into the tumor, and the other is with high. So first thing I'm going to do is to compare those two. So you can come across to this workflow, which allows me to compare experiments. There are other types of comparisons here. There are other workflows, and as you can see, all sorts of others, including drug analysis, genomics analysis, and so on. But today we're doing compare experiments. So the compare experiment workflow allows me to, you can see it's recognizing these two activated data sets, that we have almost 12,000 genes from the low infiltration and 600 from the high infiltrating data set. And I can set some of the parameters, and this is going to give me an enrichment. So compare the two data sets, enrich for pathway maps, the gene ontology processes, and so on. Now, I'm not actually going to apply this right now. It does take a little while to run, but I have one already run and ready to go, which is our next window here. So this one's actually completed the enrichment analysis, the comparison. So in orange is the low infiltrating tumor samples, and in blue the high infiltration. And so straight away you can see here is the comparison result. So this is telling me that there were 262 genes from both data sets that are common, that are either over or underexpressed in both low and high infiltrating. So these might be pathways that are CD4 specific T-cell pathways that irrespective of the level of infiltration, these pathways are running. But what's interesting here are these unique. So there are over 900 differentially expressed genes from the low infiltrating tumor T-cells, and over 300 from the high infiltrating set. So these are unique to each. And so that's where we're going to focus, is on these unique set to look at the difference between the two. Within the results, we have what's called pathway maps. So if you remember, these are the curated signaling pathways. And you can see from a quick glance here that we have enrichment in cell proliferation, in smooth muscles, enrichment in fibrosis process, in myocardial infarction, enrichment in cell matrix production, and so on. I'm not going to focus too much right now on this because these are enriched in either the shared genes or either of these two. But what's happening here is you can see, for example, with the common expressed genes, there were 35 genes on this pathway map, and there are 7 in my data set from this common differentially expressed genes that are represented on that map. So that's the enrichment of that map. Enrichment also includes these gene ontology processes. So we're beginning to get a hint here again around the cell migration concept and cell signaling, migration again. And then we have enrichment as well for processes, gene network processes being built up, and for diseases, and some statistics on the networks here. I'm going to focus today mostly on these pathway maps. I'll touch on the gene ontology processes, and we'll go on to a network. But the take-home message from this is that we have these differentially expressed genes, and I'm particularly interested in this subset here. This is coming from the tumors, from the T-cells that have a lower infiltration into the tumor. So that suggests to me there's something that's different here, that's going wrong, and I want to explore what's going on in this subset here. And I have 2 ways of exploring that. One is if I right-click here, I can sort this results specifically for this low infiltration set. And I've already got that prepared in the next tab. The other thing I can do is left-click on here, and it will build a network around this set of genes as well. And again, I've already done that. So that's what I'm going to take us through now. Okay, So just a reminder, we're focusing now specifically on the low infiltrating T cells. And so I've resorted the results to prioritize this, those that score high for this subset of genes, these 921 genes. And you can see right up, it's picked out these pathway maps to do with cytoskeleton remodeling. So you can see here 12 of the differentially expressed genes in these low infiltrating out of 36 on the entire map are differentially expressed here. There's an interesting one. This is a pathway map for immune response, and we're going to follow this one up in a moment. But you can see other processes here as well, cell adhesion, and so on. Coming down to gene ontology processes, we're looking at enrichment for the tissue development and anatomical structure, cell adhesion. So again, it's beginning to make sense, beginning to build up a story. We have something to do with cytoskeleton remodeling, cell adhesion, also something to do with the immune response. Now I'm going to pick on this one for the purposes of the rest of today's discussion, but as a biologist, I would certainly want to explore the rest of these. And particularly, you can see that of the original set, we had almost 1,200 of genes from low, 600 from the high, but you see how the enrichment has worked. We've got this map that's picked out with a huge difference between the significance for the enrichment for the low versus the high infiltrating, so the T cells. Okay, I'm going on to explore this one here, where there's a very high enrichment for the genes in the low infiltrating set for the complement pathway, alternative complement pathway. So this is what's shown here. This is one of the MetaCore pathway maps, curated pathway maps. I'll zoom out a moment just to show you the full figure, and then I'll zoom in again. So immediately you can see from the title, this is to do with immune response and specifically the alternative complement activation pathway. And look at this. In these thermometer symbols are the differentially expressed genes being highlighted for us. Red is overexpressed and blue is underexpressed. And look at this cascade down here. Every single gene overexpressed in that cascade. We also have additional ones over here, and they all lead through. So the way to interpret these maps is you start from these big in arrows. You can see that we have complement protein being overrepresented in these low infiltrating T cells, leading on through this cascade of events down through to here, where we have complement 3A, which leads to an inflammatory response. And here you can see as I hover over it, gives me a little bit more description. So that's one of the biological processes, one of the pathological processes that seems to be being activated in these tumors patients. We also have activation through here to the phagocytosis as well. So this is telling me that we have a very strong indication that this alternative complement pathway is playing some kind of role in those tumors that are having a low infiltration of these T cells. So I would want to go on and explore that and ask what's going on. How is the complement pathway related to the T cells? How is it related to the low infiltration and consequentially through to the poorer prognosis? So I would pick these as potential biomarkers of this biological pathway, of this cascade, and I would begin to explore around what's going on here in these patients. Okay. The other analysis that I did was network building, which was here. Now, this is not enrichment for a pre-built pathway map, signaling pathway cascade. This is relying on those molecular interactions in the database and enriching specifically for those interactions that are highly represented in my data set. So what I've done is built those interactions here, and it's given me a set of a list of 30 possible networks. The highest scoring network is the one here, which then is shown in this figure. And again, I've started off with a zoomed out approach, and I'll zoom in a moment, but you can see again these red symbols indicating overexpression, blue indicating underexpression, and then these highlighted arrows indicating the relevant interactions between the different components. So of either of the proteins that were in the data set, or of key components as well that that are known to interact with them. So you can see here, as I zoom in, I showed in the pathway map that we had this complement cascade of signaling through the complement system, and see again built through a different algorithm. We're seeing again reflected the same approach, that we have this complement 3 in turn being activated by this network component here, this complement 4 component, and all of the interactions that are described here either through the differentially expressed genes or through ones that are known to interact with them. And what I've done here through building this up is you can see again this cascade of activation of the complement system. But as the network is built, it's looked upstream for potential drivers. And you see what it's done here, it's picked out this one here, this GATA transcription factor, which is a key driver of all of these genes here, the expression of all of these genes here, and these in turn activate these ones here, which in turn activate, and so on. So we have here a potential upstream key hub, which is activating the complement system, which is activating the alternative complement pathway that we saw earlier in the signaling pathway. So these two approaches, the network building and the pathway enrichment, pointing to a similar story around that I certainly would be going down to investigate further. So we can begin to generate hypotheses. We have a hypothesis that the alternative complement is somehow involved in the process where we have more infiltration of the tumors by the T-cells. We have a hypothesis that GAT1 transcription factor is an upstream driver of that. It itself is not necessarily overexpressed or underexpressed from the data set, but you wouldn't expect that from a transcription factor. So I would also then begin to take this further, and then I would start my investigation of what other models are there out there that people have explored, either knockdown, knock-in, knockout of this GATA1 transcription factor. How does that relate to the immune response? How does it relate to the complement system? How does it relate to T-cells? And has it been associated with cancers before and with prognosis? So we're beginning now to use MetaCore to show potential processes that are taking place, signal cascades, potential upstream drivers, generating hypotheses of where to explore next, identifying even maybe in the future a potential target we could tweak through a drug approach to modify this cascade. All of these suggested with MetaCore here. So coming back now out of the live demo and to summarize. So let me just present this slide here. So in summary, Anne started off with a data set of 54,000 probes, very large data set, fairly quickly, straightforwardly focused in on a set of signals that were of interest to us, differentially expressed genes. She exported it from Qlucore and we uploaded it to MetaCore, and then used MetaCore to explore the underlying biology behind those variables. And using the two products in combination is a very powerful, straightforward, easy to use that enables not just statisticians and bioinformaticians, but biologists as well to explore the data behind omics experiments. And so at that point, I will stop. I've talked enough, and from there we'll take questions.
Okay, so any questions? Please use the chat to write questions to us. Do you have any? Okay, I see a question here. So it's somebody using MetaCore also wonders if it's possible to try out Qlucore. And yes, maybe if you show the next slide, Peter, there is a free trial download that you can do of Qlucore Omics Explorer, a 10-day free trial using this link here. So you're welcome to do that. And the opposite situation, Peter?
Yes, so if people want to try out MetaCore, I would ask them to contact me directly, and I will pass your details along to our local regional representative from whichever part of the world you're based, who will contact you to explore a trial of MetaCore.
Okay, and here we have another question. Do we support other data types than the array data that we just showed? And yes, that's from Qlucore side, we support a lot of different data types such as RNA-seq, proteomics, metabolomics, methylated DNA, and so on. And from the Clarivate side?
Yeah, MetaCore will handle differential express genes from whichever experimental methods you use, whether it's array or RNA-seq or so on. The files that are uploaded should include the identifier, and again, multiple platforms supported there, and it can include fold change or not and other details. Besides the transcript omics, it's also possible to use proteomics, metabolomics, and gene variant calling files as well are also supported.
Okay, great. I don't see any other questions right now. If you're interested in receiving the slides, you can request that in the little poll. So when you close this, exit this webinar, there will be a few poll questions. So if you have time to answer them, we are always very grateful. And there you can also request to get the slides. And this webinar is recorded. So once the recording is published on our webpage, we will send out an email to you so that you can download it if you like, or have a look at it again. Yes, so I think that's it for today. So we would like to thank you very much for attending this webinar, and you are very welcome to contact us if you have any questions. So thank you so much and have a nice day.